Welcome everyone to a new season of MycoTalks hosted by the MRC Center for Medical Mycology at the University of Exeter. My name is Christina Cuomo, and the co-chair today is Dr. Oliver Kurtzai. Our speakers today are Professor Angie Gelli from the University of California, Davis in the US, and Professor Tuo Wang from Michigan State University in the United States. MycoTalks are organized by the committees shown at the bottom of this slide, with online assistance in running the event from Jenny Spencer and MRC staff. MycoTalks are held at 4 p.m. UK time, usually the last Thursday of every month. And we aim to promote diversity in our field regarding career stage, gender, geographic location, and research area. All the previous talks are available on YouTube in the MRC CMM channel, and the committee is always happy to receive speaker suggestions for the future program. The format will consist of two 30-minute talks on different aspects of medical mycology, followed by a 30-minute Q&A session at the end. To submit questions, please post them in the Q&A tab, which only the moderators will see, and please specify the name of the speaker to which the question is addressed. The chairs will alternate the questions between the speakers at the end and may choose to combine questions if they cover the same topic. All the questions posted in the Q&A will also be sent to the speakers after the event, so they will still receive them even if we run out of time. The upcoming MycoTalks are listed on the website, and you must register to attend each session. The two upcoming talks for October 23rd and November 20th are listed here. You can also register to attend quarterly Myco clinics, which are a series of online talks specifically focused on clinical topics. And the next talk is highlighted here for December 11th. You can also find the schedules for Myco clinics on the MRC Center website. So now it is my great pleasure to introduce our first speaker, Angie Gelli. Dr. Gelli is a professor in the Department of Pharmacology in the School of Medicine at the University of California, Davis, where she has been a faculty member since 2003. Born and raised in Canada and an avid hockey fan, Dr. Gelli received her bachelor's and PhD from the University of Toronto and completed postdoctoral studies at Mount Sinai. Her lab investigates host pathogen interactions focused on the mechanisms driving fungal neuroinvasion. And her lab has generated assays that leveraged in vitro human models of the blood-brain barrier, including 3D organoid blood-brain barrier models, in vivo models of fungal pathogenesis to identify key proteins involved in neuroevasion of the highly neurotropic fungal pathogen Cryptococcus neoformans. Recently, her work involves the discovery and development of peptides as potent antifungal agents through combinatorial chemistry. Her research led to a patent and a prestigious research award from the Hartwell Foundation that serves as a springboard for current research on the mechanisms of blood-brain barrier dysfunction in aging, dementia, and chemical exposure. Her talk title today is Resolving Mechanisms of Fungal Translocation from Blood to Brain. Angie, we're very pleased to have you here today. Welcome you to start your talk.
Thank you so much, Christina, for that wonderful introduction. I will share my screen and get started. Great. Can you all see that? Okay. It's my pleasure to be here today. I am very excited to talk to you a little bit about the work that we do in my lab. And we study Cryptococcus neoformans. And what we find fascinating about this organism is the fact that it's so highly neurotropic. And for the past several years, we have been trying to resolve the mechanisms by which Cryptococcus can exploit mechanisms of internalization to access the central nervous system. So that's what I'll be focused on today. Okay, so you may have all seen that in the mainstream media now, finally, fungal pathogens are getting the attention they deserve. And articles in the Atlantic, Scientific America, Wall Street Journal, they are all bringing to the attention to the general public that fungal infections are a serious problem. And like the World Health Organization has recently issued a call to action, further stating that these infections cause life-threatening infections in individuals across the globe, and certainly it's about time that we start dealing with them and worrying about where we're going with these terrible infections. And the World Health Organization went as far as proposing 4 pathogens that are of particular concern. That includes Aspergillus, Candida albicans, Candida auris, and Cryptococcus neoformans. And if we look at the stats, it's pretty frightening. You probably are all aware of this. Over 6 million people a year struggle with life-threatening infections across the globe. More than 1 million people are killed every year. Thousands of Americans are hospitalized, and many millions are seen as outpatients. And so for Cryptococcus neoformans, it is the leading cause of a potentially life-threatening brain infection in immunosuppressed adults. It's also observed in individuals that are undergoing solid transplant replacement. 20 to 60% of these patients can come down with cryptococcal meningoencephalitis. And the problem is also in treated individuals. Many patients complain of long-term cognitive deficiencies. And of course, there's always the problem of a relapse. So, what is incredible about Cryptococcus neoformans is despite the fact that we've got this amazing blood-brain barrier that supports and protects our central nervous system, Cryptococcus neoformans can still invade. And so let's talk a little bit about the blood-brain barrier. What is it? So at its core, it's made up of specialized brain endothelial cells, astrocytes, and parasites. And they work together to restrict the movement across the brain endothelium. So here you're looking at a diagram, and you can see that the endothelial cells basically separate the blood from the CNS. And the parasites and the astrocytic end feet of the astrocytes work together to restrict that movement. And that's a good thing because we don't want most harmful things getting into the brain. And so if you look closely at these endothelial cells, they're specialized. They express tight junction proteins like claudin 5, occludin, and these proteins work together to maintain the endothelial cells forming this tight junctions to prevent any kind of paracellular leakage. There are also many transporters on the blood side of the endothelial cells, and they also work very hard to exclude toxic chemicals. And so together, this wonderful system protects our brain. Now, it can be bad in the sense that when we want therapeutics to get into the brain, it prevents 95% of all molecules out there from getting in. On the flip side, it protects us and it keeps our central nervous system safe. Despite all of this, there is strong evidence that Cryptococcus neoformans can cross these brain endothelium directly by a transcellular process. And that's what I'm going to focus on today. So a lot of evidence out there looking both observational studies and in vivo studies supporting this transcellular mechanism of crossing. So what you're looking at here are cryptococcal cells sitting on the surface of human brain endothelial cells. And what you'll notice is this ruffling on the surface of the membrane. And these projections from the cells can come up and completely engulf cryptococcal cells. And that suggests that there is a direct link that the endothelium is responding to cryptococcus, because if you do the same experiment with glass beads or Cephros beads, you don't see that kind of reaction by the endothelium. There have also been a lot of in vivo studies, and this is now real-time imaging that you're looking at in the panel here. And these were mice that were inoculated via tail vein with Cryptococcus neoformans. In green are the microvessels, and in red is Cryptococcus. We could see that Cryptococcus crosses the endothelium in red here directly. So part of the reason why Cryptococcus can do this is because we know that it harbors antiphagocytic abilities. And so while it can be taken up by phagocytic cells while in the bloodstream, while it's disseminating, it can also be released both lytically and non-lytically. And we believe that's also helping to drive this transcellular process. And we have also been looking at these cells as well. These are, again, brain endothelial cells exposed to Cryptococcus in green. And typically you can see stained in yellow here with phylloden, you can see this cup-like structure, same projections that we see coming up and engulfing Cryptococcus. So while there is plenty of evidence suggesting that there is an interaction between cryptococcal cells and the brain endothelium, there is evidence supporting transcellular. What we don't know is what's the molecular process of internalization. That has been poorly defined, and that's what I'm going to be talking about specifically today. And this is all very new data that we're really excited about. So first of all, an overview of our working hypothesis here and the question that we're asking, which is, what is the molecular process that is driving these brain endothelial cells to internalize crypto? Remember that brain endothelial cells aren't very happy to just be randomly absorbing and taking up all kinds of things from the bloodstream. So clearly Cryptococcus has had to adapt some processes and exploited them to be able to get in. And we know that there are two key proteins. These are both transmembrane proteins, EphA2 and CDC44. We have spent a long time looking at EphA2. What is it? It's a tyrosine kinase receptor. It's part of a large class of tyrosine kinase receptors, and it has a ligand binding domain shown here at the top, and it has two fibronectin binding domains that are known to interact with other proteins. These proteins are important. They run a whole bunch of different processes in different cells. They're phosphorylated, and they activate downstream signaling. We have shown in our studies that Cryptococcus needs EphA2 to get across in a transcellular way, and that Cryptococcus can also phosphorylate EphA2. Now, work from Ambrose Young at UCLA years ago showed that CD44, which is a glycoprotein and a receptor for hyaluronic acid, interacts with Cryptococcus. And this interaction via hyaluronic acid in the capsule of Cryptococcus can activate CD44. So what we think is happening is that Cryptococcus is somehow recruiting these two proteins, turning on a signaling pathway, and that allows it to take advantage of a type of endocytic pathway called macropinocytosis. What is it? Essentially, it's a bulk scale form of endocytosis. Now, the brain endothelium doesn't always undergo this kind of process. It happens infrequently, and yet Cryptococcus, we believe, exploits it. So macropinocytosis is distinct from other endocytic processes that are clathrin dependent and so on. It has its own signaling pathway. A lot of different proteins are involved here. It's still relatively unknown. There's still a lot of things we don't know about it, but what we do know is it's actin driven. And importantly, the macropinosomes, these large endosomes that form in the process of macropinocytosis, are large. They're in the order of anywhere between 0.5 to 10 microns. And that's important because a cryptococcal cell is actually quite large. It's anywhere between 4 to 6 microns in diameter. And some of the canonical pathways make pretty small endosomes. So we believe this is another reason why Cryptococcus may be exploiting this process. And another reason is EphA2 has been implicated in macropinocytosis. So that's our working hypothesis. That's what we believe is going on. How are we going to test it? Well, I have a very talented graduate student, Dylan Lancer, who took this project on, and we decided to develop these 3D blood-brain barrier organoids. They were developed previously. We've kind of reformulated them, and we're using them specifically for our experiments. And essentially what you do is you take primary human brain astrocytes, parasites, and brain endothelial cells. You put them all together. Of course, we need specialized plates. They have to be at a 30-degree angle. It's a little finicky. But what we find in 3 days, we can see these beautiful organoids forming. And you're looking here at the bottom of a well, and you can see them here. They're pretty similar in size, and they form pretty readily after 3 days. And what is kind of extraordinary is that these organoids will organize, the cell types will organize in the correct orientation. And what do I mean by that? I mean that the endothelial cells are on the outside, the parasites are just below, and the astrocytes fill the core. And we can look at that, as you're seeing here in these images, either live, so that we're using cell tracker dyes to look at them live, or we can fix them, section them, and then look at them that way. And so you can see that all the known markers, ZO1 is on the closer to the periphery, aquaporin N feet, right, that are part of the astrocytic N feet. Sorry, aquaporin 4 is the molecule we're looking at here. You can see the N feet are pointing away from the core. We can see the astrocytes fill the core, and we can see that claudin 5 is on the periphery. So it looks like the cell types organize as we expect them to. Now, is the organoid preventing paracellular or any kind of movement, right? We want to be sure that the integrity is intact. And so we have, or the integrity is good. And we use TEM to look at these tight junction areas in these cells. We can see them between endothelial cells here. And then again, we can look for claudin 5. We see it beautifully on the periphery. You can see this wonderful ring right around the organoid, exactly where we expect these endothelial cells to be in the organoid. And then, of course, we also look, if we take a macromolecule like fitzy-dextran, 70 kilodalton, huge molecule, which does not get across the blood-brain barrier, it shouldn't. If there's a healthy barrier, is it crossing the organoid? Turns out it is not, which tells us that the permeability is good. It's restricted. The integrity is good. If we look at FITC alone, a much smaller molecule, then we find it will infiltrate the organoid. We've also determined that the organoid is responsive to a cryptococcal cell, shown here in white. Typically, of what we've seen from our studies previously and others have shown, the blebbing, all of that we can see. If we don't expose it to crypto, we don't see that. And whether we use primary brain endothelial cells or immortalized, we see the same kind of expression of markers and function. So now that we have these organoids and the fact that they look to be like they're recapitulating, right, most features of what we expect in vivo, can we use the system in a robust manner to actually look at fungal internalization? So what you're looking at here is an organoid. This is a cross section now. Again, in red is claudin, in green is Cryptococcus neoformans. So if you look closely, here's a cryptococcal cell. We also detect a sloughing off a little bit of the capsule. That's been reported in vivo in brains of mice close to astrocytes. We kind of see the same thing in these organoids. So now what we've done is we've taken these organoids when they're in their well, exposed them to Cryptococcus neoformans, and we can see that Cryptococcus is readily internalized by these organoids. You know, we get a variability. Some will internalize a little bit more, but roughly we can see that they're getting in very nicely. We can see them here. We don't often see them right in the center of the core of the organoid. That could be because the astrocytes are packed in tightly. And what's also interesting is that if we add Saccharomyces cerevisiae, we don't see real internalization. And an unexpected result is we applied Cryptococcus deuteragadii, which is the same strain that was causing the infections in the Pacific Northwest. It's a VG2. We don't see a lot of internalization. So we think that these organoids, they're highly functional entities. They appear to be able to distinguish between neuropathogen and pathogens, neuroinvasive pathogens, and others. So we think this is going to be a suitable model for us moving forward to help us discern some of the molecular interactions that we're interested in. So in order to validate what we just observed, our organoid model, and also just to see whether these you know, what we're seeing, I should also mention one more thing. So I'm backtracking here. You'll notice that we've done these studies also in the presence of amiloride. Okay, this is an important part of this approach here. Why amiloride? Turns out that amiloride blocks macropinocytosis. It's a potent inhibitor of macropinocytosis. Remember that bulk scale form of endocytosis that we are trying to assess. Amiloride does this. It inhibits it by blocking the sodium proton antiport in these macropinosomes. It essentially prevents them from forming. And so we find that if we add amiloride to these organoids in the presence of Cryptococcus shown here in purple, we can see a significant reduction in the number of fungal cells that cross the organoid. And so that tells us potentially that amiloride here is having an effect. Macropinocytosis may be involved. To further validate that, we went back to our 2D model. This is a model that is consistently used. We used it, and we still do in the lab because it's just so useful. We take the same brain endothelial cells, we put them on this transwell here. They form a nice tight barrier. We verify that. And then we can add Cryptococcus to the top well, which would represent the brain side. And we wait until it crosses to the bottom, which is the blood side, the brain side. The top is the blood side. The bottom is the brain side. And we can collect from the bottom, we plate it, and we can quantitate it. So we have a way to quantitate crossing across the bilium in a very simple, straightforward model. And we did the same experiment, plus or minus amiloride, we can see a significant reduction in the number of Cryptococcus neoformans cells that were internalized. This concentration of amiloride does not impact cryptococcal cells. And we also did an important control here. So we're looking at transferrin crossing in this setup, in this 2D model. And why transferrin? Transferrin is a molecule that binds iron, and it has to get into the blood-brain barrier. Iron needs, the brain needs some iron, but it does so by binding to a receptor. And that process of getting transferrin across the endothelium is clathrin dependent. It should not be occurring by macropinocytosis. It's receptor mediated. And we did the same experiment. We looked at transferrin crossing in our model in the presence of amiloride shown in purple. And this is done over time. And we really see no significant difference between transferrin crossing with amiloride or without. And that's what we should expect because this is not happening by macropinocytosis. So it looks like the amiloride treatment is specifically blocking the internalization of Cryptococcus, again, suggesting that macropinocytosis here may be involved. Now, another graduate student in the lab, Amelia, she did a little more complicated experiment. And this is kind of interesting because it turns out that human cells will actually undergo macropinocytosis if they are starved of serum. And the idea is that macropinocytosis is essentially this large kind of bulk scale endocytosis where the cells could be trying to grab nutrients. And so they're just taking big gulps of the extracellular environment. And so what Amelia did is she took these brain endothelial cells, and if she serum starved them in the presence of a macromolecule, again, this is fitsidextran. So if macropinocytosis is active, a large macromolecule can be taken up. And in fact, that's what we see. The green puncta you see here is the fitsidextran getting taken up by these endothelial cells, which normally would not do that. But the serum starvation is turning on this endocytic process. Now, if we take the same cells, we don't serum starve them, but we add Cryptococcus neoformans, so now they're exposed to crypto, we see that same kind of uptake of that macromolecule. You can see here clearly by ret puncta, there's a little bit of colocalization here you can see with Cryptococcus shown in red. So we can quantitate that. And these are 3 different replicates. Again, serum starved. You can see that we see this uptake of the macromolecule without serum starving in the presence of Cryptococcus. We see that crypto is inducing that without serum starving. And then if we do the same experiment with amiloride, we lose that effect. So amiloride is preventing the uptake of that macromolecule, even though crypto is there. So further evidence, we believe that macropinocytosis may be playing a role here. Now, we also wanted to go back to determine, well, what is EPHA2 doing here? Remember, I told you that in a previous study, we found that EPHA2 activity is required for transcellular crossing. Cryptococcus can phosphorylate it, but that's really the extent of what we know at this point. We don't know exactly what it's doing and how it's promoting that internalization. And so we built these organoids expressing endothelial cells where we use CRISPR to knock out EPHA2. So you can see that the organoids form, They look pretty normal. We can see the endothelial cells lacking EphA2 on the surface. We confirmed the lack of EphA2 by Western analysis. And then we did the same kind of study where we're trying to look at the internalization of Cryptococcus neoformans. And we can see that without Cryptococcus, we don't see good internalization, right? There's significantly less internalization when we knock out EphA2. Now, what's interesting is that if the barrier had become leaky because of the lack of EphA2, you might expect that crypto would be getting in, right? Maybe by a paracellular or so on, but that's not what we're seeing. So the question was why? So we wanted to look at the astrocytes. Remember I told you at the beginning of the talk that astrocytes, parasites work with the endothelium, with the endothelial cells to maintain that integrity. And astrocytes can become active, they can induce neuroinflammation, and that can become problematic. And so we wanted to look to see what the astrocytes are doing. Are they becoming active? And one way to do that is to look at GFAP signal. And you can see that in the knockout, GFAP positive astrocytes are upregulated. We think that in this case, these astrocytes are actually working to reinforce the BBB, maybe in a compensatory mechanism, but we don't think that this is harmful. Remember, astrocytes can exist as two populations. You can have active astrocytes that are more helping to maintain the barrier. And then you've got some, you can also have another group that are more pro-inflammatory. But we believe in this case, it could be that these active astrocytes may be the A2 form, and they're actually promoting a stronger barrier. So we also wanted to know, how are these astrocytes responding to the presence of fungal cells? And so we did the same kind of study where essentially we're exposing the organoids to fungal cells, and then we're looking at the number, we can quantitate the number of astrocytes that are GFAP positive for this protein that identifies active astrocytes. And when we added Cryptococcus neoformans, we didn't see a whole lot of difference compared to control. But when we added the Gadii strain, we saw a significant uptick in active astrocytes. And that's a very interesting result for many reasons. First of all, it tells us that the organoids can differentiate kind of the nuances between these different pathogens, which is important because maybe this could be a model to take on and explore that further. It's also interesting because we think this result coincides with some of the studies that have shown that CD can promote a stronger immune response, whereas crypto is known to have to kind of dampen a neuroinflammatory response. So this needs further work to really be able to distinguish what the astrocytes are doing here. But the fact that we can see a difference and Gadii is inducing a stronger response is very interesting. So here's my model again that I've introduced previously. And so I've shown you data suggesting that EphA2, is involved in potentially driving this macropinocytosis pathway. But what is happening downstream? Now, we know that in order for macropinocytosis to happen, there has to be a lot of actin remodeling, other type of signaling has to be happening. And CDC42, which is a small GTPase, is known to play a key role in macropinocytosis. So the question is, in our system, what's happening to CDC42 when we add fungal cells to these brain endothelial cells? And so Amelia again did this study, and this is basically an ELISA. When you're looking at active CDC42, meaning has it bound GTP? When it binds GTP, it's an active protein. And so we looked at endothelial cells with EphA2, without. And of course, with EphA2 in the presence of Cryptococcus neoformans, we see this wonderful activity of CDC42. But then if we now look at the knockout, that increased activity of CDC42 is lost, suggesting that EphA2 may be mediating that signaling to CDC42, which is required for macropinocytosis. So now the question is, well, what about CDC44? Is it involved in this? So in order to test that, we decided to try a mutant of Cryptococcus. It's a CPS1 mutant that does not make hyaluronic acid. It lacks the hyaluronic acid synthase. And work from Ambrose Young's lab has shown that Cryptococcus with the hyaluronic acid in its capsule can bind CDC44. And what we see when we expose endothelial cells with EphA2 or without, we lose that activity of CDC42, suggesting that both CDC42 and EphA2 are somehow recruited by Cryptococcus neoformans. Both are required to drive this endocytosis via macropinocytosis. So, based on our data, where we see that both are required for this action to take place, we've reached out to Matthias Buck, who is an expert at Case Western University. He's an expert in modeling of these ephrin type of receptors. And so what he did is he used AlphaFold 3 to make predictions about whether there could be binding sites between CDC44 and EphA2. And in green is EphA2, in red is CDC44. And AlphaFold makes prediction about two sites. It suggests or predicts that there is potential interaction at the ligand binding domain and between the two fibronectin domains. Remember, these are both the extracellular regions of EphA2 and CDC44. And so it looks like the AlphaFold is predicting that there are two structurally distinct regions on EphA2 that may actually suggest that there's some kind of a cooperative signaling in brain endothelial cells with respect to Cryptococcus neoformans. And I also want to guide you to this glutamine 56. This residue appears to be relevant with the ligand binding domain in CDC44, and that becomes important for the next series of studies that he did. They did further analysis using the RMSD structural stability studies, and here he's looking at structural stability and binding conformations of EphA2 complexes, and he used a positive control, in this case ephrin A5, which we know is known to bind EphA2. And so basically what these studies are showing is that the lack of significant fluctuation in this RMSD data suggests that the binding modes between CD44 and EphA2 are structurally compatible. And in fact, if we look at the ephrin 5 EphA2 binding, at that same ligand binding domain, this glutamine 56 residue appears again. So it's possible that there's going to be competition between the ephrin ligands and CD44, and that may be modulating EphA2 signaling. Remember that EphA2 can undergo canonical signaling via ligand binding and non-canonical via non-ligand binding, which is what we saw with Cryptococcus. Now, the interaction at the fibronectin domain may actually help stabilize the complex, and potentially that is what's helping to facilitate the macropinocytosis. So we wanted to visualize the interaction. If AlphaFold and these studies are making predictions about potential interfaces between these two molecules, can we visually see them interact? And so for that, we used proximity ligation assays. And these are, it's a pretty powerful way to observe protein-protein interactions with fairly high specificity and sensitivity in the native environment. So these proteins are not overexpressed. This is just in situ here. And we're looking at CD44, EphA2, we have antibodies to both. And if there is an interaction that come at least within 40 nanometers or so, possibly closer, you see puncta. In this case, it's red based on the antibody, the secondary antibody we used. This red puncta would suggest that the two proteins are interacting. Now, what's interesting to us is that we see a little bit of the interaction without crypto, but when we add crypto, we actually see the interaction stimulated. We detect more quanta, more of these puncta, and we can quantitate that, and we have. And we've done this both in primary human brain endothelial cells and immortalized, and we see the same thing. It looks like Cryptococcus neoformans will stimulate this interaction, but interestingly, Gadii does not. And that kind of is consistent with what we've been seeing with our data so far. So with that, I'm going to take you back to the model and show you that what we think is going on. And so it looks like our data is suggesting that Cryptococcus neoformans is taking advantage of this bulk scale type of endocytosis called macropinocytosis. And we think it's doing it by forming a complex. It's recruiting both of these proteins that are interacting. That interaction is driving CDC42. We've shown that crypto can phosphorylate EphA2. That may be happening by PKC. This protein kinase C has actually been implicated. There is evidence in the literature that it's required for the transcellular crossing of Cryptococcus. So this PKC might be recruited, and that's what's leading to the phosphorylation. Once this large ruffles form in the membrane, they can then form these macropinosomes, take up Cryptococcus, transfer it in a transcellular way across the endothelium into the CNS, where Cryptococcus can actually survive. Gadii does not appear to induce this, and obviously there's something else going on. We don't know a whole lot about it, but that's where we are right now. There's still a lot of studies that need to really figure out all the details, but we think that this is what's going on so far. At least this is what our studies are showing. So with that, I'm going to acknowledge the people in the lab, Dylan, Amelia, Kim. They were really the instigators between a lot of these studies, and Montana, Suvita, Felicia. They started these projects, the EphA2 project, way back a couple of years ago. I want to thank Matthias Buck, our collaborator. He's been wonderful. Our image core, we've got a wonderful core facility, and certainly our funding, specifically the T32 training grants, which have been wonderful for our students. And I think with that, I will take any questions. Thank you.
Yeah, thank you very much, Angie. Fascinating talk and fascinating topics. And please, to everyone in the audience, submit your questions via the Q&A and indicate that these are questions for Angie or for our second speaker, and we will then have the discussion in the end for both talks together. So it's my pleasure now to introduce our second speaker, Professor Tuo Wang, who is actually a chemist. He studied chemistry at Nankai University in China and did his PhD at the Massachusetts Institute of Technology. And then he joined the faculty at Louisiana State University before getting to Michigan State, where he is still now as Carl H. Brubaker Professor of Chemistry. And he is working with a fascinating technology, a solid-state NMR, to investigate carbohydrate structures. And one focus of his work is actually cell wall structure of pathogenic fungi. And this includes a whole range of pathogenic fungi from Aspergillus and Candida to Crypto, Rhizopus, and Mucor species. And this is very fascinating, and I'm very much looking forward to your talk. And the stage is yours.
Yes, thank you so much for the kind introduction. Can you see my screen?
Yes, perfect.
Yeah, thank you. Yeah. So I really appreciate this opportunity to share our recent work on the use of solid-state MR spectroscopy to understand the fungal cell wall architecture, and more importantly, the adaptation to antifungal treatment. So I will give a very brief introduction, since I believe everyone here are expert with a better knowledge of the fungal pathogeneity compared to myself. So in nature, we know fungi, they are pathogens to insects, and they infect insects and digest them for nutrition, and sometimes they force them to behave strangely. And in a very famous game, one of my favorites, so The Last of Us, and also a TV series following the same name by HBO, the enemies of human society were depicted to be those fungi-controlled zombies, where depending on the level and the type of the enemies, they can have different types of fungal fruit body. Yeah, and the inspiration was largely from the cordyceps, yeah, as mentioned by the director from a recent interview. And at the beginning of this TV series, it mentions that what if, for instance, the world was slightly warmer? Now there's a reason for the fungus to evolve, and we will lose the war against them. Well, I really appreciate such a statement. I would like to point out that the director and the game developer, they didn't do a good homework, because fungal infection, they have already become a serious threat to human health, and they are threatening the lives of millions of humans each year. So we have a long history fighting against fungal infections. Back in 1950s, we have the prevalent drug MB developed, which targeted membrane for function, but also bear high toxicity. And we have nicomycin developed, which is a chitin inhibitor, but it only works on limited fungal species. And later we have the azoles, which target the extral biosynthesis for function, but they also bear high toxicity. The successful story of internal chitins, especially caspofungin and micafungin, happened after 2000, and they are inhibitors of beta-1,3 glucan synthase. And beta-1,3 glucan is an essential carbohydrate in the fungal cell wall, and the safe target has made those drugs also safe for application. But after application for 2 decades, we start to observe emerging resistance and also observing drug tolerance. So part of the reason behind that was thought to be the structural adaptation of the fungal cell wall because of the amazing structural dynamics of the microbes. And solid-state MR can help us to understand 2 types of questions. First, what's the molecular organization of fungal cell wall? And second, how does the fungus remodel its cell wall to survive through antifungal treatment? So the carbohydrates in the fungal cell wall, they are very simple in structure, considering it chemically. So chitin, they are nitrogenated sugars with the amide and the carbonyl groups that can form hydrogen bonds to stabilize the microfibril structure, and they can support the mechanical strength of the cell wall. Beta-glucans have diverse covalent linkage patterns, and they are the major building units in the cell wall, cross-linking different polysaccharides. In Aspergillus species, we have the branched beta-1,3,1,6 domain, we have the terminal beta-1,3,1,4 domain, and we have the linear beta-1,3 domain. And in Candida species and other yeast, we also have the linear beta-1,6 glucan. Alpha-1,3 glucan is a mysterious molecule whose structural function was largely unknown before, but we do think that they conceal the beta-glucans to increase fungal virulence, and our recent solid-state MR data further reveal that alpha-glucans, they serve as an important buffering molecule that can simultaneously reinforce the rigid core by interacting with chitin and generating the mobile matrix by interacting with beta-glucans. Well, we know those molecules very well. Then the next question is how those bricks are used by fungi to build the cell wall. Yeah, so and to build a dynamic cell wall, that's more important. So if we think about the biosynthesis of beta-1,3 glucan taken from this very beautiful review, we know that the beta-1,3 glucans are extruded from the FKS domain, catalytic domain of the synthase, and the shorter chains can be bridged to form longer chains and can also be decorated with side chains through the function of glycotransferase, and they can further cross-link to chitin, to mannoprotein, and even beta-1,6 glucan in, for example, in Candida species. And after 5 decades of efforts with chemical analysis, we have very good understanding with the covalent linkage pattern. We can have beta-1,3 glucan cross-linking to other beta-1,3 glucan to form larger chains and can be branched. We can have beta-1,3 glucan linked to chitin with and without mannan on the other side, and we can have it cross-linked to beta-1,3,1,4 glucan in Aspergillus species, and we can have them linked to beta-1,6 glucan in some yeast species. But if you think about the DP, the degree of polymerization, they're typically very large. Then the linkage side, they only account for a small fraction of the cell wall. Then how the cell wall is stabilized in reality is they are relying on numerous physical interactions between molecules to maintain a large integral structure. So that's where solid-state MR can contribute to visualize the structure. After 4 initial studies, we have come up with this very naive, simple cartoon illustration model of Aspergillus species. Outside of the plasma membrane, we have chitin tightly packed with alpha-1,3 glucan to form rigid cores that are both rigid and dehydrated, and they are supported by a soft matrix formed by diversely linked beta-glucans, including the linear 1,3 domain, branched 1,3,1,6 domain, and the terminal 1,3,1,4 domain. And capping the inner core is a highly mobile outer shell rich in galactomannan, GAG, which is a galactosaminogalactan, proteins, as well as a small portion of alpha-1,3 glucan. Even for coming up with such a naive model is based on extensive solid-state MR data of mobility, hydration, intermolecular packing of biomolecules with chemical analysis that details the linkage pattern and the imaging data that localize the molecules across the cell wall. After the studies of Aspergillus species, we further extend to other species. For example, one recent example includes the investigation of rhizopus and mucor species, which are important pathogens, especially for causing co-infections during the COVID. We found that those species are particularly rich in chitin and chitosan, and they are formed by 4 types of chitin and chitosan structures in the rigid core, and they are supported by soft matrix rich in beta-glucan, mannan, and polyfucols and heteroglycans, which we will show more details in later slides. And we also detail the difference between Candida albicans and Candida aureus, which has the inner and outer domain, but the polymers follow different principles to construct the cell walls of those 2 species when responding to N-channel treatment. And in another recent study, we have detailed the function of 5 types of alpha-1,3 glucans across the whole cell wall of Cryptococcus neoformans for forming the rigid core mobile matrix, interacting with chitin and chitosan, hosting the attachment of capsule on the surface, and hosting the deposition of melanin in the inner domain. So how such structural information is obtained is based on very straightforward solid-state NMR toolbox, where we first carbon-13, nitrogen-15 labeled fungi by growing them in a liquid culture with isotope-enriched media. Then we directly pack the living cells into this small container, which is called the solid-state NMR magic angle spinning rotor. So it's just a small piece of glassware that can pack materials inside without chemically perturbing them. So everything being investigated are fully native and alive. And the sample are directly put into solid-state MR spectrometer, where we can measure law to MR spectra to detail the local structural polymorphism, sub-nanometer length scale packing, hydration, motion, and domain distribution. Combining the information here, we will be able to support or exclude structural models. So the take-home message is that structural analysis of intact and living cells with atomic level resolution is now available. So our collaborator, Dr. Jean-Paul Lajit and Terry Fountain from Institute Pasteur further contributed their expertise in functional genomics, where they knock out a single carbohydrate at one time from the cell wall and let us evaluate the consequence of the mutation. And for beta-1,3 glucan, we don't have, because it's an essential carbohydrate, we don't have a mutant that can remove it completely, and the fungus can still happily grow. So we have to treat it with caspofungin to remove the majority of it. And the carbohydrate deficient mutants, such as the chitin alpha-1,3 glucan mutants, they have the corresponding carbohydrate signals depleted from the NMR spectra, as shown by the dashed line boxes when compared with the parental strain. And the caspofungin treated sample has the beta-1,3 glucan signals removed as well. So the data here has given us confidence in the resonance assignment of the NMR spectra. The treatment by caspofungin has significantly reduced the growth of the Aspergillus fumigatus mycelia, and we have chosen the 3-day-old culture to harvest because that's where we have observed sufficient difference between the drug-treated and the APO sample, and we do not need to worry about the cell lysis that will happen later. Because the caspofungin is a beta-1,3 glucan inhibitor, as expected, all the blue signals in the NMR spectrum disappeared in the drug-treated samples, and they are labeled as B, which means beta-1,3 glucan. Yeah, and here we have different carbon size showing up in the spectrum. They are all gone. And the orange signal coming from CH, which is a chitin, dominated the drug-treated sample because chitin biosynthesis has been upregulated to reinforce the cell wall mechanics in response to the stress. For the third molecule identified in the rigid core, which is alpha-1,3 glucan in green color, their original form, which is the type A here, has been fully retained in the NMR spectrum. At the same time, we have 2 new forms emerging, which is type B and type C alpha-1,3 glucans. They are not only showing up in the rigid core of the cell wall, but also showing up in the mobile matrix, which is detected through another NMR experiment, and with almost 1-to-1 ratio between the 2 new forms. So it indicates that not only chitin biosynthesis is enhanced to increase the mechanics of the cell wall, but also 2 new forms of alpha-1,3 glucans have been produced to simultaneously reinforce the rigid core and regenerating the soft matrix. And the next question we want to answer is what kind of kinetics is happening for such kind of cell wall restructuring. We have prepared 5 new samples spreading all the way from 0.5 day to 10 days. But unfortunately, as short as 5.5 days, all the beta-1,3 glucan signals disappeared already, as highlighted by the lack of intensities for the dashed line in the one-dimensional spectrum and in the two-dimensional spectrum with better resolution. So beta-1,3 glucan biosynthesis inhibition happens within 12 hours. And the quantification of the kinetics through intensity analysis allow us to understand the process better. We found that the chitin has been overproduced and reached the maxima within day 1. That allows the fungal cell wall to be rigidified to survive through the initial strike by the antifungal drug. And after that, the chitin biosynthesis has been re-equilibrated, at day 3 to reach the equilibrium, while alpha-1,3 glucan and chitosan, they have been overproduced to reinforce the flexibility of the cell wall to support growth and survival better. So this is the first time with molecular level information on the living cells that we can observe a 2-stage development restructuring process of the cell wall in response to antifungal treatment. And although the caspofungin is a beta-glucan inhibitor, we found that it's not all the beta-glucans are depleted. If we reconsider the structure of beta-1,3 glucan matrix, we know that we have linear beta-1,3 glucan domain, we have terminal beta-1,3,1,4 domain, and we have the branched beta-1,3,1,4,1,6 domain. And the branching size, which is BBR here, with the 1,3,6 linkage pattern, has rarely been observed before in any fungal species because their population within the whole cell wall is minor. However, in the drug-treated sample of Aspergillus species, we start to be able to track down the carbon connectivity of the branching point, which has similar intensity compared to the remaining 1,3 linked glucose unit, and is only present in the mobile phase and is removed from the rigid core. So this is clear evidence that we have some hyper-branched beta-1,3,1,6 glucan, where almost each single site in the backbone has been decorated by a side chain, remaining after caspofungin treatment, and is only present in the highly mobile phase of the cell wall. Another key contribution of solid-state NMR technique is the capability of resolving intermolecular interactions between molecules, which are typically revealed by hundreds of intermolecular contacts observed between different carbon and nitrogen sites across different polysaccharides. For example, here for Aspergillus, we have observed interactions in this summary among different carbons in beta-1,3 glucan, alpha-1,3 glucan, different types of chitin, chitosan, and beta-1,3,1,4 glucan. And we summarize it according to the samples, apo state, drug-treated state, or observing both. And our collaborator were able to help us visualize such interactions through the power of modeling, where they put together a large number of carbohydrate polymers in water and let them equilibrate for microseconds long, and show us how the stable interface can be formed between different polymers. For example, between alpha-1,3 glucan and chitin, and between beta-1,3,1,4 glucan and chitin. Those newly established interactions are important for stabilizing the cell wall assembly after beta-1,3 glucan is depleted. So now, although we skipped a lot of data, I think we start to have the ground to rationalize what has happened during the restructuring of Aspergillus species in response to caspofungin treatment, and how this fungus can use the carbohydrate polymers in hand to upgrade the armor, to protect itself. So after beta-1,3 glucan is removed from the rigid core, we have chitin overproduced on day 1, and also with higher level of deacetylation, which means more chitosan introduced on day 1 to day 3 time scale. And we have hyperbranched the mobile beta-1,3,1,6 glucan retained only in the mobile matrix, and we have beta-1,3,1,4 terminal domain, and we have alpha-1,3 glucan responsible for contacting chitin chitosan to stabilize the polymer interface, and we have chitosan evenly found in both rigid and mobile phase, which I don't have time to show the data, and we have 2 new forms of alpha-glucan generated to support the rigid core and regenerating the soft matrix, and we even observed the reduced content or galactosaminogalactan, which means reduce the charge state on the surface, and also fewer side chains in galactomannan, which means that the adhesive capability of the cell wall has been reduced after drug treatment. So now you can see what kind of detail, molecular level detail, we can provide using this technique to visualize the adaptation survival process and the remodeling of the carbohydrate armor for those fungal species. And it gave us a guide on how to give more effective punches to those evading microbes. And it also suggests that we need to consider this amazing structural dynamics in our development of antifungal drugs. So after the initial study of the Aspergillus species, we further extend to Candida species, especially the prevalent pathogen Candida albicans and the superfungus Candida auris, in collaboration with our mycology teams, so mycology experts across the world. And we only observed cell wall thickening in Candida albicans, but not in Candida auris when observing their cell wall thickness by TEM. And whenever we have this phenomenal difference in the phenotype, we for sure will observe difference on the molecular level through NMR assays. And as expected, we can see that Candida albicans, SC5314 strain and other model strains, they have shown significant increase in the chitin content as the first response to caspofungin treatment. And this is also observed in the Candida auris AR386 strain, which is not resistant. However, it's not observed in the I3 strain, which has fully retained its cell wall composition. And this is an antrinolkindin resistant strain provided by Fred Lammers' group and his talented co-worker Jizhou Li. And another interesting observation is that Candida albicans, they have reduced the beta-1,6 glucan content almost by half, so in response to the antifungal treatment, and this is not observed in any of the Candida auris species. And this is a clear difference between the Candida albicans and the Candida auris. And Fred's lab further constructed two mutants, KRE6A and KRE6B mutants, deleting those genes, and observed that deletion of KRE6 genes has resulted in modulated susceptibility to antrinolkindins. So it reveals that beta-1,6 glucan is uniquely important for Candida auris in antifungal response. So I will not introduce those results in detail, but you can find the details in this recent paper. But overall, the structural data showed us how those two Candida species can construct their cell walls using reserved principle, but respond to antrinolkindin treatment differently. Both of the cell wall has outer and inner layer, and with the beta-1,6 glucan bridging them, and also with chitin dispersed mainly in the inner domain initially. And after antifungal treatment, both of them increased the chitin content, and also part of the chitin has been redistributed to the surface domain and force some mannan species and beta-1,6 glucan to interact with them to create new rigid centers outside. And both of the Candida albicans and the Candida auris also decrease the hydration profile, which means the cell wall accessibility and permeability has been reduced in both species. However, they have difference. Only Candida albicans has increased the cell wall thickness, and the relative content of beta-1,6 glucan has decreased after caspofungin treatment, while it's not perturbed in the Candida auris species. And that may explain the difference between those two sisters in the Candida family. So another recent study has been applied to Rhizopus and mucor species, where we found the most beautiful MR spectra that we have collected so far, revealing 4 different subtypes of chitin and chitosan. For example, if you look at this carbon 1 to carbon 2 correlation, you can find the signal for type A, type B, type C, and type D of CS, and CS here means chitosan. And also you can find the different types for chitin. And here we have analyzed the composition of both molecules in the rigid core, and we found that the beta-1,3 glucan only account for a very small portion of the whole cell wall. And the treatment by nicomycin has selectively depleted type C chitin, which is here, and the beta glucan bonded to type C chitin and the type C chitosan, which is generated from the deacylation of type C chitin. So it reveals that the treatment by nicomycin selectively deplete a small carbohydrate complex with specific structure for the chitin and chitosan, while the other forms of chitin and chitosan, they are fully retained in the AMR spectra. That explains why the chitin inhibitor cannot be effective against those fungal species. And we have constructed a structural model where we have localized different forms of chitin and chitosan across cell wall together with beta-glucans, mannan, and polyfucrose, and also heteroglycan molecules. And also we found some protein molecules are entrapped within the chitin chitosan core and become rigidified and showing correlations with the chitin and chitosan. And here we highlighted the covalent link, the type C chitosan chitin and beta-1,3 glucan, which will be removed after nicomycin treatment. However, because the presence of the other three forms of chitin and chitosan and the increased thickness of the cell wall, the rhizopus and the mucose species can happily survive through the treatment by chitin inhibitor, although chitin is a major component of the cell wall. As you can see, the molecules in their native cellular environments, they have diverse structure. And the structure here doesn't mean chemical structure, but means conformation and interactions. So we want to understand what's the reason behind them, and we want to correlate structural polymorphism observed in our technique with the biosynthesis complexity and also functional diversity. So we started with beta-1,3 glucan, where we can resolve 3 lines, which means 3 different structures, type A, type B, and type C, for the chemically simple beta-1,3 linked glucan units. And in Candida auris, we found that type A beta-1,3 glucan is the major component in the spectrum, while type B is only a shoulder peak, but only the minor type B showing correlation with chitin, which means only type B, although it's lowly populated, is interacting with chitin, while type A doesn't. Yeah, and further analysis of chemical shifts has shown that type A beta-1,3 glucan perfectly match the chemical shifts, which means AMR signal or structure of the triple helix model, while type B doesn't. And the type B is showing correlations with chitin, and it must be perturbed within the triple helix structure, where the beta-1,3 glucan has been forced to show more flat ribbon structure when folding onto the surface of the chitin microfibrils. So that's the first clue how the chemically simple beta-1,3 glucan can adapt diverse structures. And another example is the alpha-1,3 glucans, where we observed 5 forms of them in the Cryptococcus neoformans. While if you reveal its chemical structure is so simple, it's alpha-1,3 linked, long chain glucans. But you can resolve the beautiful structural polymorphism and peak multiplicity in the MR spectra. And we found that only 2 types of alpha-1,3 glucans out of the 5 types have shown extensive correlations with aromatic signals from the melanin. And here is summarized by this intermolecular contact map showing the interactions between type A and type B alpha-1,3 glucan with different subunits in the DHI melanin in Cryptococcus neoformans. And further, our collaborators have helped us to do the haircut for the Cryptococcus neoformans cells to remove the capsule components from the cell. And this allows us to verify the signals from the cell wall and from the capsule and confirm the presence of a small portion of the capsule molecule GSM in the rigid core when interacting with alpha-1,3 glucan. For example, you can see the signals of the mannose here, and you can see signals of the xylose, both of which belonging to the GSM molecule in the capsule, and their signals are depleted in the acapsular mutant. So I will not go through the MR details here, but I want to summarize the structural concepts for the Cryptococcus neoformans study, where we have detailed the structural functions of 5 structural forms of alpha-1,3 glucan. On the surface of the cell wall, we have 2 types of alpha-1,3 glucan, which is type A and type B, responsible for interacting with the capsule and facilitating its attachment to the cell wall surface. And we have type C alpha-1,3 glucan simultaneously continuously extruding from the rigid core all the way to the mobile matrix to interact with beta-1,3 glucan and beta-1,6 glucan to form the well-hydrated and dynamic soft matrix. And we have type A and type D presumably covalent linked to each other, interacting with chitin and chitosan through physical contacts. And we also have type A and type B interacting with the melanin in the inner domain of the cell wall together with chitin and chitosan. And you can find that type A and type B, they distribute all the way across the whole cell wall of Cryptococcus neoformans. And this also explains why the cell wall of Cryptococcus neoformans is so stable, even if the chitin content in the samples analyzed by our lab is only account for a very minor fraction of the carbohydrates. So this is just a starting point. We are further trying to correlate with the structure and functional diversity with biosynthesis complexity. For example, in another recent study, we have resolved in total 45 types of chitin signals across the 7 fungal species, and we are trying to correlate them with the biosynthesis complexity of chitin molecules, because we all know that there are so many chitin synthesis families present across the different fungal species. Yeah, that's the next step we are taking. So I want to stop here. I want to summarize briefly. I hope I can convince you that technically solid-state MR and cellular MR is now ready for high-resolution analysis of carbohydrate molecules and the cell wall architecture using living cells. And the biomolecules in the native cellular context, they are very different from those purified or isolated state. They are typically highly polymorphic in structure to fulfill diverse functions, yeah, in the cell wall. And we can visualize the cell wall assembly through statistical analysis, a large MR dataset in complementation with other techniques. And biochemically, fungal cell wall is heterogeneous composite stabilized by numerous physical contacts and also stabilized by a few covalent linkages at the key points. And this composite is continuously being restructured by stress, internal and external stress, and those changes should be included in the consideration for antifungal development in the next stage. So I want to stop here. I want to thank our group members for their hard work. I want to thank our wonderful collaborators, especially Dr. Jim-Paul Lajit, who reached out to me at the beginning when we published our first study and guided me through the later studies. And he's basically my mentor and taught me everything I know about the fungi. And I want to thank the support from NIH for the fungal projects and NSF for method development. Thank you so much for your attention.
Thank you, Tuo, for that super interesting talk. We've had a lot of
Thank you.
questions come in, and we're going to alternate back and forth for you and for Angie, and start with the question for Angie. Angie, I have a question from Neil Gao, who writes great work. I was wondering whether other microbes, such as bacteria and viruses, could hitchhike on or inside or maybe connected to the Cryptococcus capsule and breach the blood-brain barrier using the CD44 EphA2 mediated mechanism. Your organoid model could presumably test this.
Yes. That's a very good question. It certainly is possible. We have not tested whether we can put other microbes on crypto to get that across, but what we have been doing is we've taken nanoformulation that are nanoparticles that are nanoformulated and carrying drugs, and we've shown that we can actually get a pretty high efficiency of crossing of these drugs in a separate project that we're doing with Jamal Lewis at Florida, but we haven't tried other pathogens. I would suspect that if we could get them to attach or adhere, it could potentially help bring them across. Certainly with these nanoparticles, that seems to be working. But with respect to other organisms, I don't know. Haven't tried it, but it is an interesting idea.
Thank you.
Right, should I continue? So I start with a question here from Neil, and this is about the technology you use. And the question is, how much does chemical fixation of the cell wall, how much would chemical fixation of the cell wall affect the spectra that you see? And Neil is thinking of a specific experiment, but I think it's a basic technical question. Could you also work with the fixation step?
Yeah, so the current samples we are working with, they are just living cells, so we need to change the samples frequently, so since we don't want to observe changes. But we have never tried fixation, but I think that's a good direction to explore. If we can show that this procedure will not change the cell wall structure significantly, then it will make the sample preparation much easier in the future. Yeah.
Okay, great. And I'll perhaps take a second question, and this is about the motive of chitin upregulation as a response, and you showed this for the echinocandins in some fungi. So do you think that this is a common theme of a stress response across different groups of fungi, or are there also groups where you didn't observe these effects?
Yeah, actually, from what we have known so far from multiple studies, this seems to be a quite conservative response, not only to antifungal drug, but also to unfavorable environment, like a hypersaline condition. Yeah, and also there are multiple principles conserved we have supported so far, including chitin upregulation, including reduction in cell wall water accessibility, and including cell thickness for antifungal drug treatment for most of them. Yeah.
Okay.
Yeah, but we of course need to extend to other fungi to check the general applicability. Yeah.
Great. Back to you, Chris.
Thank you.
I have a question from Ted White, who writes very interesting talk in my system. Do you know if azoles can enter your mature 3D organoids and treat in the organoids to eliminate Cryptococcus from the organoid? This could be important in drug.
Yes, and that is something that we are doing. So one of the projects in the lab is actually trying to see if we can develop peptides as antifungals. And we've got a couple of leads, and we have been doing that actively with these new peptides that we have to see if they can cross. And once they're in there, can they actually kill crypto? We haven't tried small molecules. In the meantime, it's all been these peptides, but that definitely is an area where these organoids can be useful. And that is trying to see how drugs are crossing across the barrier. Can we get them into the CNS? And can we show efficacy as well once they're in the organoids? So those are ongoing studies, actually. Yeah.
And then maybe we can follow up with someone who is interested in a more general question about the organoids, who said they look great. They're asking, would building them as layers rather than spheres be more like the situation in the blood-brain barrier, especially if this trans endothelial pore formation plays a role? And can you comment on that?
Yeah, that's a really good point. So the issue is the following, that the in vitro models of the BBB right now are not great. I mean, they've come a long way. And so you saw that I showed the 2D model. Now, in that 2D model where there is a plastic interface, we could put 3 cell types together so that you've got the endothelial cells, maybe the parasites underneath, and then the astrocytes on the bottom of the well. But even with that process where they are kind of layered, you still have that plastic interface there. You know, the advantage of the organoids as we see it is that the 3 cell types are directly in contact, and we're hoping to recapitulate the signaling that happens between these 3 cell types. So as of right now, building them in layers is not quite that easy because that transwell is still needed in some way. And so yeah, I don't know. We haven't tried it any other way. And so, you know, there's still a limitation. And remember too that the organoids aren't perfect, right? We can't mimic the sheer stress from the blood flow that happens in the endothelium. So there certainly are disadvantages. But, you know, I agree with that question. If we could kind of layer it up in a similar way, that would be great. But we just, we're just not there. Thank you for the question, though.
Oliver?
Right. So, Tuo, I have a question again from Neil Gao, and he owes me a beer for making me to read this one. So the question is, have you tried to look at how the NMR spectra compare for antiparallel, parallel, and mixed antiparallel, parallel forms of chitin? to get more information of how the folding affects the different capacities?
Yes, so that's a very good question. Actually, we did this in 2021 in collaboration with Tuo Wang and Tuo Wang. So where we didn't measure the other organisms, but we extracted the literature available data. Yeah, so and compared with our data across 7 fungal species. That's where we realized, okay, in the fungal cell wall, the majority of the chitin microfibrils, they are in anti-parallel way of packing. Yeah, so that's where we get this conclusion. Yeah.
Okay, and you can potentially see all the forms in a different way and discriminate them.
Yeah, and they have wild difference in their AMR signatures. Yeah.
Okay,
Yeah.
and there is another question also on the different types of chitin. Are they related to different chitin synthetases?
Yeah, that's exactly what's going on in the lab. So in collaboration with Dr. Tuo Wang, we are investigating all the chitin mutants, removing each single gene at one type. And there are some correlation. It's not all the chitin types are correlating with biosynthesis. So it's basically roughly a 50-50 contribution. 50% of the complexity comes from the biosynthesis, 50% come from structural reasons, like interactions with other molecules. Yeah, that's something ongoing in the lab. Yeah. Yeah.
Okay, great.
Thank you.
Angie, we have a question for you that also I think was maybe a big picture question moving forward from your work highlighting both neoformans and gadii. What is your thought about these different responses for neoformans and gadii in transmigration and the GFAP expression induction? Does their capsule difference? And what would that be would you hypothesize that's part of the role in these different responses in those 2 phenotypes you showed?
Yeah, and that is something that we're really interested in pursuing. I would say that there is evidence in the literature that suggests that the capsules of Cryptococcus neoformans and gadii are different, both in terms of composition. Also, the average length of the polysaccharides apparently are different. Gadii appears to have shorter GMX chains. And presumably the literature also says that that can provoke a greater immune response in some tissues. So I also wonder whether the secreted capsule components, whether they differ, and could these secreted components potentially be affecting or be responsible for the difference in response? That is totally possible. We haven't looked, but I would say that's probably a main hypothesis. There's got to be differences in the capsule. We previously showed with Cryptococcus neoformans that MPR1, it's a secreted metalloprotease that Cryptococcus secretes. We've shown that if you knock it out, it has trouble crossing the BBB. We don't know anything about MPR1 in gadii. We haven't looked. Whether it's being expressed or not there at all, I don't know. And so that could be potentially something else. So there could be something secreted, something about the capsule that's mediating these different responses. That's going to take some more work to really tease all of that out. But that's what we think could be involved here.
So I think now you've answered the second question we got, which was what types of proteins on Cryptococcus neoformans do you think are facilitating the crossing? Because you've talked both about the cell wall and about secreted proteins, but I don't know if you want to say anything more about just neoformans itself.
Yeah, no, just, besides the MPR1 protein that we identified, and it seems to be a main player, we even expressed it in Saccharomyces cerevisiae. And you saw from my data that if you try to get Saccharomyces across, it doesn't. But if we put MPR1 in Saccharomyces, we can get it to cross. So we're exploring that further to try to see exactly what is MPR1 targeting. It's a protease. We don't know what all the targets might be. Is it targeting something on the surface where it's reworking crypto potentially and making it more conducive to crossing? Or is it also and potentially targeting proteins on the surface of the endothelium? So that is an ongoing project in the lab as well.
And then lastly, related to this, are there specific factors, someone asked, that would expect macropinocytosis specifically, maybe different from, I don't know if you can separate that from crossing. Is it something secreted or something in cell wall? Everyone is asking.
Yeah, coming from the pathogen itself in terms of is that inducing the macropinocytosis? Is that the question? Yeah, you know, what's interesting about macropinocytosis, another reason why I think we're interested in pursuing this is there are other pathogens, some bacteria and some viruses use the same process. In some of those cases, it looks like it is proteins on the surface of these pathogens, different ones that are mediating that response. I don't know that it's all been worked out, but the bigger question is, could we target this mechanism of endocytosis to actually work as some kind of a prevention in getting these microorganisms from crossing? And so the fact that there is sort of this commonality among these different pathogens is interesting, but the process in terms of the specific proteins seem different for the different pathogens, but they all seem to be exploiting the same pathway.
Great, thank you. Oliver.
Right. So I'll start with this question here, Tuo, and this is, can you see something else than carbohydrates? That's the basic question. So the first part of the question is, would you, for example, be able to visualize particles like nanoparticles or vesicles go into the fungal cell wall?
Yeah, so every time when we analyze spectra, each spectra has the signals from everything in the whole container. We have not only carbohydrate region, we have lipid region, protein region, aromatics, carbonyl, and all the other molecules. They are coexisting in the same spectrum, but their signals are dispersed, so they're separated from each other. From our initial studies, we are only focused on the carbohydrate components. Yeah, but later with more time and manpower, we may be able to analyze the other signals that has not been analyzed in the previous studies. But the quick answer is yes, we can analyze them. Yeah.
Okay, so I guess so there are questions regarding nucleic acids in the cell wall. There are questions about for example, parts of the Cryptococcus capsule in the cell wall. This could be seen. And
yeah,
how about the dynamic processes when you get let's say, a nanoparticle attaching to the cell wall, then migrating through and fusing with the membrane or something like this?
Yeah, and if we want, we can further add a label to the nanoparticle, any type of label, then we can see them clearly. Yeah.
Okay, so I see a lot of more high impact publications to come from this. And then I have another question, and this is, what about subtle differences? So did you, for example, compare the action of different echinocandins? So basically all of them working on the same target molecule, but perhaps with slightly different kinetics, or did you compare amphotericin and nystatin or different azoles with regard to their action?
Yes, so for the echinocandins, we have provided a supplementary information figure in the 2024 Aspergillus study. So where we show that the three echinocandin drugs, they have quite similar behavior regarding the MR spectral side. Yeah, so but for the other drugs, we didn't have the opportunity to look at them yet. Yeah, that's where this platform is important for facilitating future collaborations. Yeah.
Right. Christina.
I have a question, Angie, from someone also thinking about the why of this is happening. Does Cryptococcus naturally cross the blood-brain barrier? And what are the factors that are driving it to do so? Are they related to survival, to nutrient acquisition, or to something else? Why does it preferentially cross the blood-brain barrier and invade the brain?
Yeah, so that question comes up a lot. And it's interesting, right? Of all the places that it can go, why is it so neurotropic? Why does it like the brain? And there are some ideas out there for why this might be. So first of all, you know that Cryptococcus makes melanin, and that is supposed to help it survive some of the onslaught of the host trying to kill it. And so the idea is that the brain is full of something like dopamine, it's a catecholamine, and that is a precursor for melanin production. So one of the ideas is, well, maybe it likes the CNS because of that. Another point is that the spinal fluid, the CSF, is full of proteins and peptides, and potentially it's using it for nutrition as a nitrogen source or a carbon source. That's a possibility. There's also a lot of inositol in the brain, and that could be another reason. We know that there is a study showing that inositol can actually improve or promote its virulence. So I think those are some of the reasons, and certainly getting out of the bloodstream is probably what Cryptococcus wants to do. If it does escape the monocytes, obviously where else can it go so that it doesn't get destroyed? And perhaps the brain does provide some kind of a safe haven, given that maybe the immune response is not quite as vigorous. And I would say, I think those may be some reasons that might address why crypto seeks the central nervous system.
Okay, thank you. Oliver?
Right. So Tuo, there was a question in the Q&A about the potential covalent linkages between different cell wall carbohydrates, and if you can say something about how these covalent linkages are actually synthesized.
Yeah, so I have limited knowledge in this aspect, but my knowledge mainly from, like, for example, Jim Paul, and his colleagues who did the chemical analysis of the alkali soluble, alkali insoluble fractions of the cell wall, and then analyze their covalent linkage patterns across the polymers. Yeah, so that's where they found the mannan to beta-glucan to the chitin complex, and also beta-glucan to chitin complex. Yeah, so I think those are the work from the field, so in earlier studies. Yeah, and for the enzyme part, sorry, I don't have knowledge in that.
No worries. So, and another question that, excuse me, this is a typical medical question, but we have this beta-D-glucan as a serological marker, and we know it's positive for Candida, but it's not for Mucorales. And you explained nicely that the Mucorales have a far lower content of the beta-D-glucan in the cell wall. So, do you think that this is by itself sufficient to explain the lack of usability of this test, or is it also a different way of how the beta-D-glucan is arranged in a cell wall that contributes to these fungi not secreting this molecule?
Yeah, so most of the time we found that it's not a simply composition issue. Yeah, so it might be more or less how those molecules are accessible or not, what kind of role are they important or not in the cell wall, so for function. Yeah, so for example, alpha glucan is like has like a very high abundance in many cell walls, but turned out that they are not like very interesting for antifungal development. Yeah, so basically they are playing like a buffering molecule role. So wherever it's needed, so they will be there. Yeah, so that may explain to that answer indirectly. So I think both the composition and also their structural function should be considered. Yeah.
Right. And there is one quick question just to be very sure. So this is about, do you analyze intact cells or extracted cell walls? I think you were mainly talking about intact cells. Would it potentially also be possible to analyze cell wall extracts for their composition?
Yeah, so currently we are analyzing mainly like living cells. So it's basically everything that can be put in that small container. They can be kept in their native state and then analyzed. Yeah.
Okay.
Yeah.
Great. So some final questions, Christina, from you.
Yes, there's a question about the organoids and the molecules that they take up. How was it defined that they were only able to transfer fungi-like size macromolecules and not larger? Is it possible to understand the kind of optimal size of microorganisms that can be taken up by the organoids, maybe using the beads that you were showing, I think, in some of them?
Yeah, so if the endothelium is intact, right, clearly it's not going to take up large molecules. They would have to be probably below 500 Dalton. At least that's what's been shown in vivo, right? Molecules that are below 500 Daltons can readily get into the BBB. So something like crypto, which is huge, obviously on its own, it's not going to just freely get in unless, of course, we're talking about the specific mechanism. But if it's some other large molecule, it won't get in. So, if something larger, a larger molecule is getting across or a bead or whatever, it would mean that the BBB is becoming permeable, whether it's paracellular or it's damaged or whatever. So, because by definition, anything larger than 500 Daltons should not be getting in. In fact, that's what you would want, right? You want that barrier intact. So that would be tricky to do unless you specifically damage the BBB to get these larger things in. I'm not sure if that answers it, but yeah. It's all about maintaining that permeability, right?
Right.
Yeah.
So I think this concludes our session. Thank you very much to two very engaging and exciting speakers. Also, thanks to the audience for all the questions coming in. And the final words is a reminder that the next MycoTalks is going to be on 23rd of October, and we hope to see all of you again. Thank you very much to everyone, and have a nice evening or a nice day.
Thank you.
Thank you.
Thank you.